Thank you, Lisa. It's great to be honored and asked to come and give a talk today. I was asked what type of topic I would possibly want to speak about and what we'd be going over, and I thought about various different things I worked on over the years. I originally started my work working with bats and then transitioned from bats to arachnids and doing the ectoparasites of bats, and then studying mites and ticks and other little creepy crawly things. And while I find them absolutely fascinating, most people just look at me with a vague look and wonder what is wrong with me when I talk about mites. So I was like, that might not be the best community talk to do. So the other thing I spent a lot of time working on is I've done a lot of work with pollination ecology and also biocontrols and looking at various different things that we can use to help control and try to remediate some of the things in the environment that have gotten a little out of whack. So that's what we're going to be talking about today. We're going to be talking about the kind of adventures I've gone through over the past, at this point, 16 some odd years, studying and working with biocontrols of saltcedar. So as the talk's entitled here, Beetles, Weevils, and Scales, oh my, it's because we started out with tamarisk leaf beetles and then somehow started dealing with tamarisk weevils, and then we had tamarisk scales show up on the side as well. So we've had a variety of different biocontrols come into the picture, and we've been working, tracking, and monitoring their activities, how they've been affecting saltcedar, and how the changes in saltcedar has affected the entire ecosystem and community along the Rio Grande. So that's what we're going to be talking about and going through today. Now, as we get into this, the first thing I'd like to kind of point out is that I'm the person presenting this talk, but this is not a one-person show. This has been the work of many different people and many different agencies involved. When we first started our project out, it was referred to as the La Junta Project, which began back in 2006, and it was a collaborative effort to try to get these beetles brought in and established into the forgotten river stretch of the Rio Grande to try to control this saltcedar menace. We had work with the Rio Grande Institute, the USDA ARS agency, NRCS, and the Chihuahuan Desert RC&D group. Now, these groups all were working together, and in particular, NRCS worked with us to help put this project together. Now, this project is based on trying to control saltcedar. Now, saltcedar in the genus Tamarix is found throughout the world in many different locations. In particular, it primarily came from the Middle Eastern, Turkey, and over toward India. Those are kind of the main hotspots of where it originated. And then from there, it migrated, it dispersed, it moved about through the environment. Saltcedar is very good at going everywhere and getting itself well established. As you look at how saltcedar spread throughout Europe and the Old World, we have it moving over into China, we have it spreading through parts of Africa, we have it going throughout large parts of the Mediterranean. So this has been known to be a spreader, a problem, an issue. So how did it get to North America? Last time I checked, there's this large ocean called the Atlantic in the way. How did it get there? We did it. We brought this thing in back around 1823 and started actively planting saltcedar for the novelty of stream bank stabilization. We don't want to have our stream banks get eroded. We want to put this stuff in to stabilize it and to keep those things from eroding and washing out. Well, it did a darn good job of that because it stabilized the stream banks, consumed the stream beds, and then spread down and up the stream beds. So we had this huge monoculture grow and develop over years to the point where this nice little picture of the Pecos River around Iraan, Texas, that was taken back in '71. You can sort of see a little bit of the Pecos River in the middle there. The rest of it is just walls of saltcedar. This stuff really did kind of take over the area. When you look at the map that's illustrated here, you can see that, quite frankly, most of the main rivers of the southwestern United States have been impacted by saltcedar. This stuff has pretty much gotten everywhere. So it's gotten to be a big issue. It chokes out waterways, it dominates the agricultural area, and it makes it so very few other things can exist, persist, and grow. Ecosystems are based on diversity. If you don't have diversity of plants, you won't have diversity of animals. You don't have diversity of insects, birds, mammals, you name it. So we were basically changing the environment into this giant farm-like saltcedar plantation running along every riverbed. Not the ideal of what we're looking for. So what had happened is USDA ARS worked with various other researchers and tried to identify what sorts of beetles we could use to, what sort of insects we could use to control this pest or menace of a salt cedar plant. Now, as we look here, this is looking at the southwestern part of the United States, and it's looking at several different species. It's looking at technically 4 different species of salt cedar beetle and where we've been working to release them. Before we started our project, there had been work already done in Colorado and even already in Texas around the Big Spring area to try to work and establish these beetles to control and eat salt cedar. Now, the La Junta project itself was specifically started as a grant to the NRCS that we were applying to get money to be able to establish these beetles on private property initially for the primary purpose of reducing the salt cedar pressure on the ecosystem, and in particular on the waterways, to increase grazing for ranchers. So this was not initially a water reclamation for wildlife or for ourselves. This was initially a ranching grant to try to get water for animals so we could get more grass growing for them to feed on. Because if you have a field area full of salt cedar, your cattle are not going to graze very well. They're not happy. They don't want to eat salt cedar. They want to eat grass and various other plants. So the whole point of this project was to try to open up these areas and reduce the amount of salt cedar. The grant that we got working with NRCS started the funding back in July of 2006, and we also got some additional funding from the Meadows Foundation, the World Wildlife Fund, and the Rio Grande Institute. So all of these groups were working together with us to try to make this possible. I was representing Saul Ross's biology department at the time. We also had members of USDA ARS, so the Agricultural Research Service end of the U.S. Department of Agriculture. We had people from IBWC, so the International Boundary and Water Commission, because we were working along the Rio Grande, and last time I checked, that's an international boundary, so we need to be working with the politics of the situation, the Texas Cooperative Extension, and Texas Parks and Wildlife. So there's a lot of people involved with this project to get things started. Maybe? Going the other way. There we go. So the initial objectives and goals that we had for this project, we started with 3 basic goals back in 2006. The goals that we had were first and foremost establish 3 sites along the Rio Grande between Presidio and Candelaria in this forgotten river stretch, the part of the river that nobody seems to be caring about or acknowledges even exists. We then were going to work with 3 different species, or at that time ecotypes, because they weren't recognized as species yet, of Diabria leaf beetle, to see which ones were the best ones suited for areas, which ones would establish, be able to grow, propagate, and actually do the job. And then we also planned on doing overwintering study work to see if they'd be able to survive out here. Toward that goal, we had to go forth, locate various different sites that would be suitable for growing, rearing, and ultimately releasing these beetles. We had to establish cages over salt cedars so we'd have a controlled cage environment so we'd be able to keep them in control and know what's going on. Within those cages, we had sleeve environments, or ultimately loose in the cage beetles, and then we'd monitor the growth and development of those populations within those cage environments. Once numbers got high enough and we had permission, the goal was to be able to release them from these areas, monitor beetle numbers and activities wild in the field, and then also monitor vegetation and vertebrate presence to see what's going on in our treated areas versus the original unmodified salt cedar areas. So this was the original kind of plan that we had going forward. So for our initial sites, we have illustrated and shown down here the city of Presidio. Up here we have Candelaria, we'll call that a town. Anyone who's been to Candelaria knows that it's a very, very, very small community that you can go to. It's a beautiful place, but there's not a lot there. We worked with various ranchers, and Armendariz, Munez, and Prieto. Those individuals all agreed to allow us access to their ranches to start this project, as well as Rancho Pensada. We had 2 sites, one on each side of Candelaria, that we were allowed to set up and establish tent sites. So we were planning on setting up 3 initial sites. We're starting with 5. We're already getting a little aggressive and trying to see what we can do, because the more things you get set up, the better chance something will have to be established. So we go forth and we need to figure out which ecotypes or species we want to introduce. So here's a map of the old world where saltcedar is found, and here's the location of these various different beetles and what they do. So they're all diabria beetles, but there are different species, sublineata, elongata, crenata. All of these are different species found in different parts of the world. Now, the subtropical or Tunisian beetle was considered to be probably a very good ecological match for the desert of West Texas. So getting in the Chihuahuan Desert and this part of Texas, we figured that getting kind of this Tunisian effect is probably going to work quite well, because if they can handle living in that sort of dry, hot climate, they should handle being out here pretty well also. We also looked at the Mediterranean beetle or the Crete beetle, because that seemed to also do well in fairly dry, modulating hot environments. The larger beetle was found from Uzbekistan, which we nicknamed the Uzbek beetle. The Uzbek beetle is a larger beetle, and we were really optimistic about that beetle because it is a larger beetle. Larger beetles are capable of eating more and also laying more eggs. So we're thinking, ooh, this one will be better at building up numbers and spreading in the environment because it's naturally a better egg producer. That's what we're going to shoot for. These other two species are just ecologically not fitting the environment quite right. We didn't even want to touch them. If anything, crenulata, the northern species, does great up in Colorado, but it would do horribly down here in West Texas. So we kind of picked our big three and focused with that. Now, in terms of the beetles themselves, when you start out with the beetles, they start out in the egg stage, this nice little budding thing that looks like a little tiny cob of corn stuck on the side of a saltcedar. Those are their egg masses. They then have the first and second instar larval stages, which are pretty much jet black and fairly small and indistinct. Then we get to the third larval stage where they actually start getting a little bit of coloring and start actually looking like the adult beetle, at least in terms of coloration, but still a blackish, long, grubby-like beetle. It forms a yellow pupal casing, which it goes through its growth and development for metamorphosis, and then comes out as one of these adult species. Either our northern, Mediterranean, large, or subtropical. And I have stars next to these three because these are the three that we were planning on introducing at our cage sites. So each one of those initial sites, Armendariz, Munez, and Prieto, we designed and set up these cages, and we're going to introduce one of each one of these beetles and get them established and get them started. We got everything set up. We started that establishment process. We started looking at them growing, developing inside the cage. And then, unfortunately, life is full of chaos and misconceptions, and things go wrong. And this was no exception to that. We got a call from our group that we were working with in New Mexico who had collected the beetles in the various countries in Europe, brought them in, had them in quarantine, raised them in isolation, reared them so they would be able to grow and develop and have pure colony strains for us to be able to release and work with these individual ecotypes or species as our permit was given to us by the U.S. government. The Uzbek beetle. We found out that those were not pure Uzbek beetles. They had had an accident in the laboratory. They were accidentally cross-contaminated with the Mediterranean or elongata beetle, and therefore they were all hybrids. So we had a whole bunch of hybrids in a cage. We couldn't release the hybrids because our permit wasn't for hybridization. If they hybridize in the field on their own, that's fine, but we couldn't start with hybrids. So we started the project up in 2006, 2007, got everything set up, had all three cages ready and running, and then we had to go fumigate and kill all of them and reset the project all over again. So 2007, 2008, let's do it right this time and make sure everything is not crossbred or hybridized. And we can start working on just doing the elongata or the Crete beetle and sublineata, the Tunisian beetle. We're not even looking at the Uzbek beetle anymore. We washed our hands of that mess because we couldn't get pure strains. So we're just focusing on these two species of beetle, and we're beginning to introduce them along this stretch. For the Crete beetle, we worked on introducing them at one site in Big Bend National Park. That was actually set up at St. Helena Canyon, was the site that we had set up for the Crete beetle. And then the Tunisian beetles we set up around Gravel Pit Adams Ranch area, as well as working along the stretch here of the Forgotten River stretch, and then near Candelaria at Rancho Pensado 1 and 2 is where we also released more of these Crete elongata beetles. So we basically had elongata on the ends and then sublineata or Tunisia beetles in the middle. So we're still getting two different types. We'll look and see how they're doing and be able to compare how they grow and develop. So first things first, now that we're doing the restart, we really figured out how to get these things set up and establish our cages, because sadly we wasted a year doing a lot of work before we had to reset. So we talked to the ranchers, found a good site that had some good salt cedar, picked a good area that was nice and congested, and then went about setting up our cages. First thing we did was cut and trim a salt cedar so it would fit inside one of these cages. They were metal frame cages that we put insect draped cloth over the top of. We actually dug and trenched 6 inches down all the way around for the frames so that these things would actually be sunk and buried in the ground. So there would be no way the beetles could get out of the cage until we were ready to let them out. And then because we were working with ranchers and we were working on functioning active ranches, once we got our cages set up, we then erected cattle panels around the tent cages. Because the thing we found out from the first year is that after we fumigated, we were going to go back and collect all of our equipment, and some of the cows decided that those are really good scratching posts and therefore knocked some of our stuff down. So we learned, okay, put cattle posts up and make sure you can keep the cattle out of the site this time. So we worked out those bugs and kinks and we were ready to go. Once we had the cages set up, it was time to start introducing beetles. The first thing we did for control is we would take the beetles we received from the laboratory in New Mexico where they've been reared, and we would put them into these gauzy sleeves. The sleeves were designed to keep them tied and anchored onto a branch of salt cedar so they have plenty of food to eat, but to also keep them together. The neat thing about these beetles is as these beetles feed and start to go into reproductive-like behavior, they release pheromones. And some of the pheromones they release are aggregation pheromones designed to draw them in together and to keep them all into one good tight group. We didn't want the beetles just to start dispersing and not finding each other. We wanted to encourage them to be able to breed. So to do that, we just kept them all close and snug together in these bags inside the cages, so there was nowhere they could go. They would have no option. They'd just eat and breed. That's all we wanted them to do, and it worked pretty well. Their numbers actually started to increase pretty impressively. Within 2 weeks, we already saw some noticeable change. Within a month and a half, we started having to take them off the sleeves or take the branch, cut that branch, relocate the sleeve onto another branch with the dead branch in it, and continue the process. After a few goes of this, we just gave up with the sleeves and started to let them loose in the cage itself. Once they were loose in the cage, then we had the monitoring part begin. Our monitoring consisted of going through these trees and checking the branches and conducting 2-minute branch counts. Basically, you start working on a branch toward the base, work your way out toward the tip, and you have 2 minutes to count all of the eggs, larvae, adults that you can find present. 2 minutes doesn't seem like a long time, but when you're counting these things, it feels like it's forever. And you're sitting here with multiple tally counters frantically trying to record and document all these different things that you're finding here and figure out how your stages are coming along. So we're looking here and readily seeing those nice good egg clusters, occasionally L1 and L2 larvae, the presence of those nice third star larvae that are really eating and consuming. This is the longest lived larval stage, and then the adult beetles either individually or when they're in copulation. So lots and lots of good beetle activity at this time. Working with IBWC and CMEX, we were able to get permission from the U.S. government and the Mexican government to be able to go forward with our release. The U.S. government was fully on board with this, otherwise they would have not funded and granted and allowed us to start this project to begin with. The Mexican government was not quite as happy and convenient with wanting to go forward with this, and we had several rounds of, no, you can't release yet, no, you can't release yet, and then eventually we got okay, we're not going to tell you you can't release, but we're also not going to tell you that we're going to support this project. And we went, what does that mean? Is that a yes or a no? And IBWC said, let her rip. So we went, okay, they didn't blatantly say no, off with the cages. So we took the cages off, opened them up. Here's a picture of Tara Pulaski opening up one of the cages for us, and you can see all the beetles inside as they were being released out in the environment. Now they've been building up in here, they've been saturating the area with their pheromones. Hopefully they won't disperse and go very far, they'll stay together as a group. Well, we got lucky. That's exactly what they did. We went out, started doing these branch counts, looking for them in the neighboring areas, and would see them on the surrounding saltcedar bushes. And when they got out there in numbers. They would literally be dripping larvae off of these saltcedar bushes. So they were occurring in high numbers, huge amounts of these things, indicating high levels of reproduction and just prolific breeders. We noticed that they would go from the cage itself and hit a neighboring tree, and then once they heavily defoliated and hit that pretty hard, they'd go and hit the next one. And they were literally just leapfrogging from tree to tree to tree, beginning to work and actively defoliate. Now, this took us a while to get to this stage. We were at this point in about 2009, 2010, to beginning to see them moving and beginning to really get established and start working on eating saltcedar in the open, as we're seeing here. And it worked pretty well, to the point where we started getting situations like this, where we can clearly see our cage that's been opened up, and this entire area is completely covered with saltcedar that has been defoliated. We know it's defoliated because it's not a bright, vivid green color anymore. You can now see the red of the stems of the trees. So this lets you know that most of the green has been eaten off, and the beetles have had a field day here. So they're doing great. So by the end of 2009, 2010, we kind of reestablished what we were doing and tried to kind of see where we stood. So the initial objective, set up 3 sites along the Rio Grande. Well, we actually started with 5, so that didn't hold for terribly long. And it continued to grow and develop to the point we ended up at that point with 14 sites running from Candelaria to Big Bend National Park. So we were actively working that whole stretch, got lots of beetles active and present that we were monitoring. We wanted to see which of the 3 subspecies or species would work best in the area. We figured out the Uzbek beetles were not successful. We worked with the Tunisian and the Crete beetles, and we had the Crete beetles at a couple locations, but they weren't doing terribly well. They were able to kind of hold their ground. The Tunisian beetle, Sublineata, was doing quite well, and it was actually beginning to grow and expand its range. So that's what we started to really focus on, is who's actually expanding and growing, who's going to get established and spread like an invasive species, except this time it's to control another invasive, and hopefully it's just going to hit what we need it to hit. And that was the whole point of what we're trying to do here. As far as the overwintering study, I'm not going to cover that. That was kind of a cute little study where we basically put a whole bunch of cups in sand and put substrate in the cups and put the cups outside and put them inside the cages. And when it got cold, we put beetles in the cups and let them go into a torpor diapause. And then once a month, we drive out to all these sites, pick up a couple of these cups, go sit in the truck with the heater on and let it warm up enough to see if the beetles would move around. Okay, it's not dead yet. Put it back in the cold. And that was literally what we did for 2 years, every month during the winter. So kind of a boring study, something that we ultimately couldn't even publish because there was just not enough to it. But it was still something to get them established. Once they were doing this in the wild on their own, we didn't care. They were good to go. So we have this illustration here of the La Junta district showing this kind of range of the Rio Grande that's largely ignored and not given the credit it's due. And then by 2010, we can see we had beetles established or present throughout most of this range. So we were pretty excited about how things were going along. We were getting lots of good beetle activity, and they were even moving up the Rio Conchos, invading and attacking saltcedar in Mexico as well. So focusing just on our stretch here and looking at Big Bend Ranch State Park, we could see that we had some good activity around Madera Canyon and Lajitas. We also saw some really good activity around Redford. Alameda Marsh was one of our big hot spots, and then going all the way up toward Candelaria. Now, as far as the defoliation went, we started with a tree, and then another tree, then another tree, and then a little patch. That was up through 2009. In 2009, we finally had our kind of breakthrough moment. We had beetles actually kind of really growing, establishing, and spreading an area. And this was the Tunisian beetle. So we saw that we started out here. This was about June of 2009, some good activity covering a nice little area. We then increased the polygon a little bit by August, and by September, October, this had gotten even bigger. If you look at our scale here, this is about 100 by about 75 meters. So it's a pretty decent little range. It's not huge. We're not talking about acres upon acres upon acres of land, but they were beginning to get well established. So we were getting very happy. We had a Saltcedar Beetle meeting that folks from Colorado, Utah, and all had come out to visit us. They came down to this site, and everybody oohed and awed and was so happy about how well Ruidosa was doing. So we were excited. We had lots of promise for what was going to happen for the future, and then the future happened. Does this sound like someone's going to pull the rug out from underneath us? Yep. Come on. You can advance. There we go. Go back one, please. In March of 2010, there was a fire on the Mexican side of the river right across from Ruidosa, and it spread across the river and got our site and burned our site to the ground. When you look at this, there had been cages that had equipment strapped to those cages. All of that incinerated and was destroyed, and all the salt cedar burned up, and the ground looked like a moonscape. So we're like, well, so much for our beetles, because last time I checked, our beetles were in the ground when this happened. You get high heat sweep over them, and they probably all pop like popcorn. So we didn't have any beetles left at this site. We were really disappointed and going, well, that was our establishment. That's another 3 or 4 years down the drain. I guess we have to start over again. Well, there's a thing here. We're dealing with salt cedar. Salt cedar is extremely persistent, nasty stuff. Just because you burn it at high temperatures doesn't mean it's dead, it's just annoyed. And therefore, this is the picture from March. Come on. Here's September. That's not very much time. Look how much stem basal growth is growing on these salt cedar plants. They're just bushing right out again. And the neat thing is all the salt cedar growth you see here is new, only a couple months old. So it's new, it's young, it's actually somewhat succulent, and it doesn't have a lot of the high chemical concentrations you normally associate with established mature salt cedar. So things can eat it. Things will readily eat it. So we're going, okay, we've got a really good field of tasty salt cedar. If only we had beetles. But we did have some beetles at another site. They picked up on the scent and the smell of this salt cedar, and they moved in. And as of November 2010, they found this nice little burned patch of regrowth salt cedar and literally chewed every single bit of it to nothingness. And we went, oh, they're back, we're good. And the project was still going and we were still running forward. So we were very excited. It's like, okay, we're still in the running, the project's still going. Where they came from was from Alameda Creek. Alameda Creek meets the Rio Grande just on the east side of Presidio, and this turned out to be one of our hot spots. In 2009, it also had a nice little patch of habitat. It had about 2 acres worth of salt cedar they had defoliated, but it wasn't quite as impressive as the Ruidosa site. Then in 2010, that's when they kind of took off. In April of 2010, it had expanded to the yellow polygon here, which was about 7 acres. 2 acres to 7 acres is a rather dramatic increase. That's a 3 times increase right there in area that they had completely defoliated and stripped the salt cedars down. And that is looking at this at the time of April. Now let's look at June of 2010. That's 2 months later. In 2 months later, everything that's outlined in green is where these beetles were defoliating. Please note, they have absolutely no respect for international borders. They were actively defoliating in Mexico too. They didn't care because they're beetles. So we had them defoliating from 7 acres to 51 acres in 2 months. And they kept growing. We had hit the magic point where the beetles' aggregation pheromones were holding them together. They were breeding. They basically saw unlimited food availability, and therefore they just started to breed and increase and breed and increase and breed and increase and exploded in population. We saw these populations move and spread and start moving from Alameda downstream toward Big Bend National Park ultimately, and upstream, eventually reaching all the way to Candelaria and beyond. So this is when it really took off. So we were really happy. Things were working. Everything was great. Mostly. There were still some problems. There were some challenges we were facing. Some of the biggest challenges we had were flooding. When you look at the life stages of these beetles, the eggs are laid up in the salt cedar. The larvae are crawling around up in the salt cedar, eating and nibbling on those leaves and sucking the juices out of them. The pupal stage, the chrysalis effectively, just like a butterfly, the pupal stage doesn't move. It's not anchored to anything solid. It's going to drop to the ground, pupate on the ground, and sit on the ground until it's ready to hatch out as an adult and then get back up into the shrub again. That works great when there's dry ground. However, if you take these wonderful little beetles and you flood the area, they have a problem. Pupae don't swim. They also don't snorkel very well either, apparently. So we lost sometimes entire generations of these that you'd have them grow, get to the third larval stage, they're ready to pupate, they drop to the ground, bloop, bloop, and they're gone. And then you're like, well, so much for the June crop. I guess we have to wait and see what happens for the next couple months for another group to come through. There were times the flooding was so severe we'd be parking 1/2 to 3/4 of a mile away from our sites, putting on waders and hiking, mushing our way in here just to try to do branch counts on the adults and the larvae that were present, and then slug back out of here again. It was a pretty challenging experience, and pointed out that flooding is definitely going to be something that could have some long-term effects on moderating and keeping control of this population. The other thing we had was we had predators. One of the biggest predatory issues we had was actually ants. Ants will come in here and ants will feed on anything that they can find. They are going to eat plant material, they're going to eat protein-based animal material. And what these ants found out very quickly is that L1 and L2 larvae are small, squiggly, and don't put up much of a fight. Pupae don't put up any fight. So you'd sometimes see rows of ants leaving one of the saltcedar bushes we were working on. You could just see the row of them, all of them carrying a pupae above their heads with their mouth parts as they were marching along going, thanks for the treats. When we were first trying to get these established, this actually got to be a problem where we were actually having the ants taking away more of our beetles than were actually being produced. We additionally set up some surveys, putting some pan traps out there to see how the ants were and how abundant they were. In areas where it was just getting bad, we actually had to break down and draw them in to kill them in controlled traps. The controlled traps we used were using max force ant bait inside pan traps to draw them in, and we put covered pan traps out in the environment to draw these ants in. Now, we had to have some sort of bait to really get them in, because most insects aren't just going to come to a pesticide. So we used the elaborate, expensive trap of really cheap hot dogs and just cut up little bits of hot dogs, threw them in these pan traps, let them bake out there in the West Texas sun. That produced a smell, and boy, did it draw ants in. And the ants would come in, eat the hot dogs that were coated in poison, and therefore it helped knock the ant populations down. We only had to do this for about 2 years. Once the beetles really started producing in numbers, there were more beetles than all the ants could possibly eat, and we were okay. On a lesser scale, we also had some issues with spiders coming in. We'd see them feeding on the beetles, but nothing enough to worry about too much, as well as some assassin bugs coming in and feeding, particularly on larvae. So when we originally got set up, we had our 3 sites that ultimately then turned into 5 sites, and as of 2015, we had our 14 sites set up, 13, 14 sites running, and pretty much continuous beetle presence along this entire stretch. The other thing to note is that at this point, we're only saying sublineata. Where did those nice little yellow dots go? There's still yellow dots here, indicating that the Mediterranean beetle, the creep beetle, is still present. Well, as time passed, they would be identifiable and recognizable at those sites only, and completely surrounding them would be the Tunisian beetles. And then we stopped seeing really good creep beetles. We started seeing things that looked kind of like a creep beetle, but kind of like a Tunisian beetle. We did a couple collections on those, examined the genitalia, which is one of the ways you tell these beetles apart, by collecting them, pinning them, taking the genitalia, turning it inside out, looking at it under a microscope, and examining the shape. Not necessarily an easy, fun thing to do. The shape didn't match up to either one of them. It was an intermediate. Okay, this isn't fun. Let's go to the wonderful world of genetics and let's do some DNA work on them. Oh, they are definitely interbreeding. They hybridized. We now, at this point, do still have the Mediterranean creep beetle's presence in the area. It's just about 25% of the genome that's left that shows up in these various individuals. So this population is going strong. It is pretty much all sublineata with a good mixture of the Mediterranean beetle's genes mixed in there just to kind of give them some flavor as they continue onward. They pretty much got ecologically absorbed. In 2011 and 12, we had really good beetle activity. The work from 2010 kept exploding. It kept going great, and we had these huge broad areas showing lots and lots of dead saltcedar. Now, the neat thing about how this worked is that the beetles would defoliate the saltcedar and strip all the leaves off. Saltcedar is a stubborn, resilient plant. It just pushes leaves out again. So the beetles eat that round of leaves. And they would do this 4 or 5 times per year. They would defoliate and strip the trees down. And then they would do this for year after year after year. By about the 4th to 5th year of this defoliation process, guess what? Even saltcedar eventually dies. So we started actually getting some dieback and seeing some actual saltcedar death after about 4 to 5 years of continuous defoliation. By 2014 and 15, we started seeing unstable population numbers of beetles. And we also started seeing that there wasn't as much defoliation taking place. The reason why, most of the defoliation had already taken place. A lot of the saltcedar had been knocked back. There wasn't as much food available. You can't maintain a horde of saltcedar consuming insects if there isn't a whole lot of saltcedar to feed them. Well, where was the saltcedar? Well, there's saltcedar in other parts of the river, and they could smell and detect it, and they started moving upriver, following the saltcedar and moving as a wave northward. We continued to work and look and sample. We found a few areas where you'd actually get some good high beetle numbers and intensity, but it was only small isolated patches that were doing this. The majority of it was fairly low beetle intensity. This low defoliation would sometimes only be 3 to 5% defoliation, as opposed to 80, 95, and 100% we were seeing earlier. So the beetles really had kind of largely moved out of the area, and they were moving, in this case, north, heading toward New Mexico. We also started seeing these other 2 biocontrols showing up in the area, and I'll talk about them more when I get to them here in a little bit. So 2016, we started seeing more beetles showing up, and the biocontrols got a little bit more aggressive. By letting the saltcedar kind of rebound and grow back for a couple years, those saltcedars that survived, it started to provide enough ample food again for the beetles to come back and start to actively feed on it. This is actually something we were looking forward to, hoping and praying was going to happen. The idea of using a biocontrol is that you are not actively applying it when you need it. It should get established in the environment, it should maintain itself, and it should ultimately function in some sort of cycle. The saltcedar will get pushed back, the beetle numbers will drop off. As saltcedar rebounds, the beetles should also rebound as well and recover in a delayed fashion following the saltcedar. This is what this illustrated. So we started seeing this cyclic process where the saltcedar would go down, the beetles would decrease, saltcedar would grow back for a year or 2, and then you get the beetles coming back again. In 2017, we once again saw isolated patches of beetles, but a lot of low intensity throughout the general area. But we did see some heavier beetle activity in the northern parts of the Rio Grande, particularly getting into New Mexico. We had gone past El Paso and we were now getting into southern New Mexico, going up the Rio Grande and up the Pecos and hitting both of those areas. So we were beginning to see a lot more beetle activity moving throughout those regions. So here's a map from 2017 showing the beetle activity. And we can see our area down here where all of our dots were really these 2011, 2012 activities in terms of when they showed up. And then we see that in 2017, these things were moving from El Paso all the way up past Albuquerque. In 2014 and 15, we were seeing them moving up the Pecos as well. So we had a lot of really good activity, but it was all moving north. They were all continuing to follow this major river system. In 2018 and 19, we continued to see some beetle activity, but just not uniform heavy numbers, just periodic numbers. But as you can see here, they were occurring in some good high patches in places. One thing that was interesting, we found some pretty good numbers here near the Black Gap Wildlife Management Area. Okay, so they're also moving downstream, moving toward Amistad. We also found them in the northern reaches of New Mexico. I actually did a family vacation trip up to Albuquerque, and being a good scientist and biologist, when you go on a family vacation, you periodically sneak away and you do research. And I went and visited all the different state parks and places I could get down the river and collected and looked at beetle activity, much to the dismay of my family. So they were not entirely thrilled I turned into a research trip. I was like, oh, those saltcedars are redding over there. There's probably beetle activity. Let's go check. Again. Okay, so we went, checked, and we found them all over the place in that area. So they really were doing well. In 2020 through 2023, I just did not have the funding to be able to keep things up. I also was dealing with various family issues in terms of health issues. So I wasn't able to survey as much as I had been, but I still did some surveying in 22 and 23 and found beetles at Terlingua, Lajitas, Alameda Creek, Presidio, as well as periodically along Highway 170 between Presidio and Candelaria. So we did have a fair amount of continued beetle activity. Even to today, they're still present, they're still working, they're still helping to reduce saltcedar. Important thing to remember with a biocontrol is not going to eradicate your problem. It doesn't go in and kill all of it. That's not the point. The point is to push it back, reduce its numbers, reduce its intensity so that other things can come in and produce a more balanced ecosystem. We're never getting rid of saltcedar. Saltcedar is here to stay. It's like getting rid of cockroaches. It's not going to happen. We can't get rid of all of them. But what we can do is push it back enough to give everything else a fighting chance. This is the current map put together by River's Edge, which used to be known as the Tamarisk Coalition. And this shows the map of the western United States, and all these various different dots and shapes here are saltcedar activity and monitoring. We see our nice activity area down here. We see the active current monitoring where they're going nuts, sampling, collecting, and working along New Mexico, along the Rio Grande, working all the way from basically El Paso up to Albuquerque. We see some older work that had been done on the Pecos, and then we see Colorado, Utah, and Arizona still being some pretty big hotspots for beetle activity and study. So we're still seeing people working with the beetle. We're still working on trying to deal with this. We're just having to adjust and focus on other issues. New Mexico had a big focus they had to shift with in the fact that New Mexico is breeding territory for the southwestern willow flycatcher. That bird is a federally endangered species that lives in willow trees. Unfortunately, willow trees have largely been wiped out because saltcedar replaced them. So what do the birds do? They adapted. They started to breed in saltcedar. So when the beetles come through and defoliate the saltcedar, now you have an endangered bird that's built a nest that is nice and out in the open, exposed to the sun, getting nice and hot and toasty to bake their young to a nice golden tan, as well as to serve them up as a serving platform for any hawk flying overhead. Not ideal. So we've been dealing with the repercussions of having to deal with the USDA with this biocontrol program and Fish and Wildlife for the southwestern willow flycatchers, and they've been arguing over whose fault it is and how to fix it. And the end solution was plant some willow trees, darn it. And they started working on mitigation. Unfortunately, this did take something like 5 years to come to this assessment after arguing. So we're beginning to get mitigation work going on. We're getting things to start moving forward and bouncing back. The next thing we have here is talking about these other two biocontrols. I'm just going to briefly introduce them. The first one is the splendid Tamarisk weevil. This was a really neat little critter that my grad student Allie and I came across back in 2015. We saw this on saltcedar and said, "What the heck are you?" This gorgeous, beautiful little specimen we came across. Didn't know what the heck it was at first. We saw pupas. We then went back, collected, observed them again, got some adults, collected active pupae in September of 2015, and then reared that pupa until it hatched out to an adult to get a perfect, pristine adult and to be able to work on identification. So here's what we had in the laboratory in September. We had this pupal casing that literally looks like somebody blew a bubble and then made it aerated with this beautiful hardened net-like material, and the pupa is located inside that, rattling around like a little bean inside a cage. This is what it looks like when it's out in the field, stuck to the side of saltcedar, and this is the weevil that emerges from it. This little guy will take his mouth parts, nibble on the saltcedar, and suck and draw fluids out and cause damage to the plant. We got these saltcedar weevils actually causing up to 30%, sometimes 40 or 50% defoliation damage to those plants. So it did some pretty good work for us. We collected these around Colorado Canyon and Big Run Ranch State Park initially, and then found out through looking at records that they had been found up in the Panhandle initially, as well as Nevada, Oklahoma, Utah, California, and Mexico. As far as we can figure out, our population probably traveled across through Mexico and jumped back up north into Texas, and then has been spreading along the river and thus getting themselves fairly well established in the region. In terms of where we found these things, we found them along the Rio Grande. We found them as well going all the way up to Lake Balmorhea and even over toward La Mesa, Texas. We have also been able to continue to study and collect these things, and as 2018 shows, we found them in Midland County, Valverde County, Reeves and Ward County, Pecos County, Crane County, and Crockett County. Basically, we went and followed the Pecos River, and anywhere the Pecos River crossed a road, we'd get out, pull off on the side of the road, crawl down under the culvert bridge, jump somebody's fence probably, and then go and look for weevils, and we'd find weevils. So these things were getting all over the place, really good numbers. As you can see, cute little guys, beautiful patterning, and then a really good picture of that pupal casing with the pupa inside that lattice work. So just some really fun, beautiful specimens we collected. The other thing that we found was a tamarack scale. The tamarack scale would literally just start covering the salt cedar plants and making it look like someone had dipped it in frost. So you'd see all these little bitty white dots stuck to the tamaracks. Tamaracks has very small, tiny leaves as it is. Each one of these basically is covering one of those little leaflets. These things would get up to about 30% defoliation on an individual salt cedar, and they would generally cause a little mild inconvenience for the salt cedar, generally weakening it a little bit, but generally not doing massive defoliation. The other thing that we noticed about these things is that one of our native ladybird beetles, the twice-stabbed ladybird beetle, apparently thinks these things are cotton candy. And whenever we'd find these, you just look around a little bit, you start seeing huge swarms of these black with 2 red dot ladybird beetles just sitting there and hoovering these things up. So apparently they were good food for our native ladybirds. And the downside of that is they're not going to be a good biocontrol if they're getting constantly hammered by our local predators. So they're present, they might be doing something, but we also generally did not see them doing a great job by themselves. As far as the scale was going, we did find them along the Rio Grande in various different locations. We also did see them up in Pecos County, Crane County, and even found them in Eddy County in New Mexico next to Lake Brantley. So if you go up to Carlsbad and you go to the state park just north of Carlsbad, we found them up there as well. So they are throughout the region as well. So this is a map just kind of showing where these things were found, showing the scale and the weevils, just basically blanketing the entire Big Bend area. We even got reports of them over at Del Rio, dealing with Lake Amistad. We had a rancher from Lake Amistad who collected something off salt cedar and sent it to me and said, "What the heck is this thing?" I was like, "Oh, weevil." So we got one from there. And then, as you can see, we had some good activity along the Pecos. Balmorhea Lake, we had some good presence. That Carlsbad location, and then La Mesa, for some reason, we're also even getting them all the way up there, both scale and the weevil. So this has shown a lot of different types of biodiversity in terms of these biocontrols and how they're working to hit and knock salt cedar back. So the end result of this is we've ended up with a lot of standing dead salt cedar. That's kind of what we're hoping to see. There is some living salt cedar, but it's far reduced in biomass. It's not the dominant monoculture it used to be. It's allowing other things to have a chance to grow and propagate and have their impacts and the effects in the environment as well. So we need to look at and monitor and see what's going on with that. Toward that end, I've had various different graduate and undergraduate student projects that have been working on trying to assess what's going on and what the impact of this biocontrol has been. So I'm just going to give you very short synopsis, like 1 slide or so each for each of the grad students and undergrad projects to show that this has actually had an impact on the environment. First one I have here is Ali Hassenfluh's study. She was looking at the impact of salt cedar control and the presence of salt cedar and how it's going to impact the different types of insects present. Now, this is a group of all various different insect orders: the Coleoptera, the beetles, the Hymenopterans, the bees, wasps, and ants, Lepidoptera being your butterflies and moths, Diptera being your flies. And on each one of these, they're graphed by the different species of plants we found them associated with and the numbers collected. When you look at Coleoptera, salt cedar is in orange, and the only thing that Coleoptera is dominating on is Chrysomelidae. That's the only thing that we saw large numbers of beetles on. I wonder what beetle these were. Oh, that's right, they were our leaf beetles we released as a biocontrol. So if you excluded that, their number would be down here. So basically it showed we increased the numbers of one beetle, but everything else, generally as you look at this diagram, the other colors are higher than the orange, indicating that these other plants are actually better for the environment for insect diversity. For Hymenoptera, Apidae, your honeybees did great because they're generalists. They don't care. They will use anything. They're little nectar and pollen, insert bad word here, to get access to everything. So as a result, we're going to see lots and lots of bee activity because they don't care. They'll use anything. They're very broad generalists. For halictid, some of our sweat bees, there's some generalists within there. They'll use anything that's sweet, so they're going to come in pretty good numbers. But also within the halictids, you can see they really loved getting into the mesquite and the desert willows. Those are actually just as good or better. For megachilids, your cutter bees, they really like tree tobacco. Our vespids liked desert willows and tree tobacco. For our dipterans here, salt cedar, we see cucilids. Here, this is going to be dealing with your mosquitoes. Are your mosquitoes picky in what sugar water they drink? No, they just want sugar water. So they were pretty abundant, but everybody else was generally more abundant in other plants. For Lepidoptera, for some reason, Ali switched from orange to green for this one, but salt cedar is low, actually doing pretty good for nymphalids and doing a little bit better for pyrrhids. These are more generalized types of Lepidoptera. We're seeing a lot of the Geolectidaes, however, are predominantly found on other species of plants. So the other plants are providing greater diversity. When we look at salt cedar versus the native plant communities, we see that, go back. Get a little happy there with me. Come on. Say, go back and stay there. Okay. No touching. If we look at the Shannon-Wiener Index of Diversity, I hate you. Stop that. Hands off. Okay. So for the diversity index, it's looking to see how diverse the ecosystem is. If we look, the salt cedar community had a 2.55 diversity. The other mixed community of plants, 2.96. Other plants provide a greater diversity of organisms that are going to be present in them. More insects were found in that environment. So we definitely see that the other communities are doing pretty well in terms of salt cedar versus native plant communities. The insects like other plants. They like the diversity to interact with. Another student who did their thesis, it's on the table that time. Okay. Karen Nielsen, she collected her data back in 2010, 2011, some early work when we were still working on managed versus controlled type environments. When we look at her diversities for mammals in non-native environment, i.e. salt cedar, 2.7. You look at mammal species diversity in the native, 5.9, twice as large. Mammals seem to like native vegetation. The herpetofauna, your snakes, toads, turtles, etc., we have a 1.8 for salt cedar, a 1 for native, and a 2 for mixed. They seem to kind of not really care too much. They were kind of low diversity in general as a whole. When you look at your birds, 2.2 was our species diversity for birds and salt cedar, 4.0 in native vegetation. Once again, almost doubling. So this really helped to illustrate the idea that birds and mammals prefer a diverse native habitat as opposed to a salt cedar monoculture. Much more diversity, more individuals present. Getting into the undergraduate work, we saw insect diversity. Stop that. Okay, it's just getting overzealous here. We saw insect diversity according to salt cedar controlled areas versus still being managed areas. The areas that were controlled and had more native vegetation that had come back in and had kind of grown and mixed into the area, as I show with the red boxes over there, those areas were statistically significant compared to the areas that were still actively being controlled where salt cedar was trying to be pushed back. When you got salt cedar under control and you allowed native vegetation from the seed bank to start coming in, we got a lot more diversity of insects. What's driving that diversity of insects? That's what her second year project was, and it was looking at the plant diversity, and we see the exact same pattern and trend. Stop that. Showing that plant diversity also was significantly higher in the controlled areas than it was in the areas where we're still trying to control and mitigate for salt cedar. So this showed the beetles were having an impact. They were knocking salt cedar back, and they were letting other things come in to get established to provide more plant diversity that led to more insect diversity that led to increased bird and mammal diversity. So are we helping the ecosystem to kind of bounce back from the impacts of salt cedar? Yes, it has actually had a good, strong effect. And it has worked well. It has controlled salt cedar. It is a very restrictive, controlled biocontrol. It doesn't eat a lot of other things. It does not eat corn, it does not eat cotton, it does not cause other problems or threats. There is only one sister species that it will sometimes nibble on, and that is athel trees. So, athel is not native. Athel is an exotic. It was something that was brought in here. So, when we look at athel trees, athels have a much looser leaf structure network. They're a more billowy plant. They also tend to be a lot bigger. They grow to really full-size trees. You get good 50, 60-foot tall trees out of these things. The original lab work that was done showed that the beetles far and away preferred saltcedar for breeding, but they would potentially be found on athel and basically refused to even do anything on Frankenthenia. So we're thinking, okay, we should be okay. They shouldn't really have any problem and get established on athel trees. So that was the original thought. In July of 2010, we were doing a monitoring trip and we stopped at one of our sites along the way, and on our way to a site, we noticed that there was some saltcedar growing up along the side of the road that was being defoliated, so we stopped and looked at it outside an RV park, Loma Paloma. And we got there, we saw this nice defoliated saltcedar, and right next to it is a little athel tree growing. Who looks healthy? Athel looks healthy, saltcedar is getting chewed up. All right, that's going well. It also, I would like to point out that according to the literature, athel trees have to be planted. They don't propagate on their own. Bull. They propagate by seed just fine. So, they don't like athel trees. Well, unfortunately, when you've got a horde of several million of these beetles moving across the landscape, they're hungry and they eat everything they can actually eat. So they will start to eat the athel trees. This created a huge hubbub. I got lots of wonderful hate calls from Presidio of people telling me I was killing their shade trees, I was killing these trees, I was making poor children go to school and get heat exhaustion because the trees around their school was being impacted. All these horrible things because the saltcedar beetles were eating athel trees. Now they did do this defoliation. They went and chewed their way up a fair distance up the road and managed to get almost halfway up from Presidio toward Candelaria. Now, in September, we saw significant regrowth coming back. We had told the landowners, do not cut down your athel trees. They're not dead. Just wait a little bit. They're going to leaf back out. The beetles would have moved on. It'll be okay. And sure enough, the plants grew back, and we worked actively with the Texas Forestry Service to help explain things and try to get as much information out as possible to tell people it's not a disaster. It'll be okay. So in July, we have some pretty nasty defoliation. By November, that looks pretty healthy again. They're fine. The plants are fine. Just leave them alone. The beetles have moved on. Everything's great. Everything's honky-dory dandy. Nothing could go wrong. Oh, that's right. 2011 was the year of the freeze, wasn't it? Boom. Everything gets hit. We get blamed for our beetle activity weakening the athels and causing them to be susceptible for the freeze. We had to go and collect data from Rancho Pensado, where we had athel trees up there that had never seen beetles, and compared them to beetles located down here around Loma Paloma and such like that. So we have northern, never seen a beetle, southern, had been seeing beetles. Both of those look pretty equally unhappy. The freeze was pretty detrimental. So, February. Here's that nice freeze damage. The trees are pretty much toast, and everybody's unhappy. But the beetles are kind of dead too because they didn't handle the freeze terribly well. It took them a bit to bounce back. So let's go and look and see what's going on. Oh, the trees are bouncing back. 2 or 3 years later, they had dead crowns from the branches that did die off, but in general, as you can see here, the athel trees are doing quite fine, even in the presence of continued beetles. It's only when the beetles are huge high numbers that they'll even nibble on athel, and even then they move on as quick as they can because it just doesn't taste as good for them. It's not as palatable. They can't lay eggs on them as well. That's the fundamental problem. So we got them to kind of move on and keep going. So where does this leave us? Well, this leaves us with the fact that research is never done, questions are always out there, and you have to keep working on stuff. We've continued to do occasional trips as we've been able to pull off and do branch counts as we can and try to record where the beetles are, what they're doing, and how they're interacting with the environment. I'm hoping to get some additional funding to be able to get a student to be able to help me with this project again and to continue to work and learn more about the beetle activity along the Rio Grande. We're also actively working with folks in the Pecos River area to try to see about getting these beetles pushed and get a new active reintroduction of them going in the Pecos region as well. So we're trying to get things going to see how well we can keep this going for the future. Lastly, I'd like to wrap up acknowledging all the folks who helped throw money our way over the years. Sul Ross State University has provided some faculty enhancement grants over the years. NRCS and the Soil Conservation Service got this all started, along with CDRI and the Rio Grande Institute. The SAWC was involved with providing several grants through the Rio Grande Research Center for us in the middle years of the project. And then the final big years of the project, when we were working under funding, was funded through the National Park Service and the World Wildlife Fund. So we've gotten a lot of funding over the years to keep this project going. We've also worked with a lot of people over the years. Like I said, this is not one person, this is a team, a huge squad of people. The Beetle crew, including the late Jack DeLoach, who really was the man who got this project started. I had a great pleasure working with Jack DeLoach. I actually had academic history and connection with him. Dr. Jack DeLoach had been the major advisor of Dr. Jim Richardson, who was the entomologist at Sul Ross back in the late 90s and 2000s, and he was actually my major advisor during my master's work. So it was neat to get to work with effectively an academic grandfather on this project. James Tracy went on, got his PhD at Texas A&M, and is the person who's been doing the genetic work on these beetles. Alan Knutson works with Texas AgriLife and is continuing to do some work with Saltcedar. Mark McGee, Ty Fan, Mark Dene, they've all either moved on or passed on in various ways. And then Anne-Marie Hilscher, still located and working with us at Sul Ross, was instrumental in some of the earlier work on this project. Worked with numerous students over the years. Andrew Berzin, Tara Pulaski, Kara Nielsen, and Ali Hassenflew were students specifically focusing and working on this grant project. Also had numerous undergrad students and McNair students who worked on these projects over the years. And then lastly, friends and family here at Sul Ross in the area who've helped me to be able to put the time and effort into working on all these things, learning as much as I can, and generally just listening to me babble about beetles, insects, and other strange, crazy things as I get fascinated with the world around me. So with that, thank you all very much, and I will entertain any questions you have at this time.